Hi, students. We are in part 2 of chapter 10 on biotechnology. Some of the main tools that biotechnology and molecular biology especially uses is the techniques that we developed to isolate DNA first. Isolating DNA, the genetic blueprint of life, is actually very simple. It can happen in 2 steps. So because DNA is protected inside the nucleus, so you have to break the cell wall and cell membrane first, if it's a plant, and then you have to break the nuclear membrane in order to get to the DNA. So the first step in DNA extraction would be to break open the cells. And in the lab, it's so simple to break the cells. We just pulverize it, grind it up in a pestle and mortar, and we add a detergent. Like even Joy detergent works well. And that will break the cell membranes. The second step would be to separate the DNA from all the other cellular debris, mainly proteins. And then you precipitate the DNA with some alcohol. So after we filter the extract, we would add cold isopropyl alcohol, and that will pull out the DNA. It's called precipitating the DNA, and you can actually spool it in a glass rod. It's so fascinating. So DNA isolation is pretty simple. You break the cell membranes first, and then you would precipitate the DNA. And then by doing the precipitation a few more times, you can clean it up. And then you can quantify the DNA using a spectrometer. So that we have mastered that technique now. So let's move on to the next technique. Gel electrophoresis. You must have done it in the virtual labs. Gel. So there are 2 words here. Gel is referring to a semi-solid jello-like material. It's called agar. It's a seaweed. It's translucent. So they will pour the gel, and in the gel, there are these holes called wells where you can load the sample. DNA is negatively charged. So when you load the DNA and pass an electric current, DNA will move towards the positive pole. And the separation of the DNA particles will depend on its charge and size. The big molecules will sit on top of the gel, and the smaller molecules will move further down the gel. That's why it's called gel electro. That means you're using electricity to separate the different sized pieces of DNA. So now this is a very, very valuable tool for a molecular biologist. And then they will stain the DNA, using maybe ethidium bromide, or there are other dyes that will light up the different bands of DNA. The first well usually have something called ladder, and then that is like an internal standard. And it's this banding pattern that is used in DNA fingerprinting to solve crime scenes. Another huge discovery is the discovery of restriction enzymes. So these enzymes were first discovered in bacteria. See, because bacteria doesn't have an immune system like us, so in order to protect itself, it has these enzymes that will chop up the virus that's infecting the bacteria. And that's how it was discovered. So in molecular biology, restriction enzymes are like a scissors. They call it the molecular scissors. So, and here's a nice picture on the right here. So this enzyme will look for a specific sequence. See, in this case, it's looking for a specific GAATTC sequence. And the enzyme is cutting it. And because it doesn't cut through straight, it has overlaps. So these are called sticky ends. And this is desired in molecular biology. Because of this overlap is a very good thing. And now, because you're able to cut the DNA, you can insert a foreign piece of DNA shown in green here and stick it back together by an enzyme called ligase. So DNA ligase is a molecular glue. And restriction enzymes are molecular scissors. And here's some examples. EcoR1 was discovered in Escherichia coli bacteria, the E. coli bacteria that lives in our gut. And this enzyme is going to look for this recognition site, GAATTC. So now we are able to cut DNA at a specific sequence. It's very targeted. And we also have an enzyme called DNA ligase that will help to seal the cut back together. So you can insert a foreign piece of DNA and glue it back together. So now this is what has given rise to recombinant DNA technology. Another technique, very cool technique, was this discovery happened in the Yellowstone National Park. So they have discovered a bacteria. It's called Thermus aquaticus. And it has a DNA polymerase that is functional at a very high temperature. So this instrument you're seeing here is called as a thermocycler. So anytime you want to amplify DNA, so for example, you want to amplify this segment of DNA, if you heat it, the hydrogen bonds between the double-stranded DNA will break. It's called DNA melting. So now you have two strands of DNA. And then using this DNA polymerase that can work in very high temperature, you can synthesize the complementary strand. And then you heat it again. You melt it and anneal it, melt it, anneal it. So like if you do this like 21 cycles and so forth, you can amplify a segment of DNA, make many, many copies of the same DNA. So this is a very valuable tool that molecular biologists use. This also requires a primer. Okay, they will add a small primer and then DNA polymerase will do complementary base pairing. See this slide explaining what I just said. So it is a very good study slide for you that you can look at later. Another technique is polymerase chain reaction. Not polymerase chain reaction. What am I saying? Restriction fragment length polymorphism, RFLP. So in this technique, what they'll do is they will chop up the DNA using restriction enzymes. So the cellular DNA will be isolated and restriction enzymes will chop up the DNA and then it will be run on a gel in order to form a DNA fingerprint. And there's a few other steps here. But this has provided to be a very useful tool, especially for DNA fingerprinting in forensic science. This is the most important picture. I really wish you understand the beauty and the importance of how recombinant. Recombinant means DNA from two different sources. And again, they have some technology to make this happen. So if you take a bacterial cell, which is a prokaryote, it has some extracellular DNA. It is in the circular DNA. So it is called as a plasmid. And here is the gene of interest. Let's say it is the insulin gene. Okay, you want to make a lot of insulin for people suffering from type 1 diabetes. So let's say this is the human insulin gene. Okay, so you cut out the gene. So you have the gene of interest. You have to put it into this plasmid first. So that's why it's called as a vector. So you use the same enzyme to cut the plasmid and also to cut the gene of interest. And you glue that gene into this vector called plasmid. So now we have the insulin gene in the plasmid. And then when you put it back in the DNA, you can make several copies of the plasmid with the insulin gene. And when the bacteria grows again and again and again, it will make a lot of insulin that you can filter and make a medicine of. So recombinant DNA technology is used to make the proteins of interest, like insulin, human growth hormone, some peptides that are medicines in human disorders, or you can put it in the plant itself. For example, the corn plant that we see here in New England, They have the pesticide resistant gene and the herbicide resistant gene in the plant itself. So when the insect eats the plant, the insect will die because the pesticide gene is in the plant itself. So this is the GMO food that we are very common in. We have high yielding corn, disease resistant corn, salinity resistant corn, short duration corn, etc., etc. And then in bioremediation, we have oil eating bacteria. And, we saw industry, how they have some enzymes that makes our clothes so soft. And we have a lot of medicines that are made in bacteria now. So this is the overall picture of recombinant DNA technology. Again, I wrote out everything I said in words so that you can look at it later. And this is one big application of DNA sequencing in forensic science. So here, by the banding pattern of DNA in a gel electrophoresis, you can place the suspect in the crime scene. How do you get a recombinant DNA into a plant? There are two main techniques in agriculture that they use. One is using a bacteria called Agrobacterium, and the other one is called as a gene gun. Gene gun. So they will coat the recombinant DNA on the pellet. Actually, the pellet is made up of like gold or something like that, expensive. So here you can see how the plant cell is grown in a culture. It's called a scalus. They'll take the plant cells and they'll grow it in a petri dish. It'll form a callus. And then what they'll do is they will project the DNA into the scalus, thereby creating a transgenic plant. It's kind of random, but it does have some success rates. The next technique is the more powerful technique. Here you would, in this case, they are trying to put a herbicide resistant gene into a plant. Why do you need a herbicide resistant gene? So that when you spray the herbicide, only the weed will die. Your plant will not die, because it's resistant to that chemical. So here they will take the gene of interest, first clone it into a bacteria first, put it in a plasmid and in a bacteria. Here the bacteria is called Agrobacterium. Agrobacterium is a bacteria that forms tumors in plants. So this Agrobacterium bacteria has evolved to infect plant cells. And that's how you introduce into the plant, thereby making a GMO plant. See, if you're doing genetics the old-fashioned way, in order to transfer a gene from one crop to the other crop, it will take about 15 to 20 years. Here, you can accomplish this in 5 weeks. That's the beauty of technology. Okay, I already said this. The gene gun, I told you just now. The DNA is coated in the gene particles and it is shot right into the callus or the plant. So now you all should be familiar with this pros and cons of genetic engineering. Environmental wise, if the pesticide gene and the herbicide gene is in the plant itself, it is very good for the environment. You're not polluting the environment because the plant is so resistant to drought and so forth. You can, reduce tillage and, turning over the dirt, which causes soil erosion. You can produce high yield in plants, and you can produce pharmaceuticals in bacteria and so forth. So it has a lot of cons. Food is so cheap nowadays because of genetically modified organisms. The harvest is so rich, like tenfold more harvest. However, there are, of course, a lot of disadvantages that, there are activists who talk against GMO foods. They talk about how genes from the GMO crop can migrate to the wild type. There's a lack of biodiversity because we are growing the same species over and over again. And also some, it's not written here, but there's lots of papers that talk about allergies and, human GI problems digestion problems that can happen. So there are both pros and cons for GMOs. Another main thing I want you all to be aware of and appreciate would be the Human Genome Project. So it took 3 companies nearly 13 years to accomplish this, and they completed sequencing the genome in 2003. And again, DNA sequencing can be complicated, but again, there are many techniques, but the most popular one is the automated technique, where they will, melt the DNA, and then they will chop up the DNA first, and then they'll melt it, and then they will add nucleotides that have a fluorescent dye attached to it. And it will terminate after it comes to that specific nucleotide. And then depending on the banding pattern, you'll be able to read the alphabets of the genome. So now the whole human genome has been sequenced. So now what? So what? Why is it important? It is a very big diagnostic tool. If you are prone for breast cancer, your genes will tell it. There's many diagnostic kits that you can buy. Not buy, you can send out your DNA, your spit, and they will tell you if you are at risk for getting breast cancer, ovarian cancer, and so forth. So be aware the DNA is sequenced. So now, then of course, they have also, Josiah's presentation on CRISPR, which is clustered regularly interspaced short palindromic repeats. So that is the expansion of what CRISPR technology is. But what you have to know is we have the tools to cut and paste DNA at a very specific region. In other words, you can cut out the mutated or the damaged DNA and insert the correct copy. So the success rate is higher. So again, it gives hope. For genetic disorders, there's kind of no hope, but now it gives hope. So this is like one of the biggest technologies that's available now.